I'm so glad you found your way here tonight. Every night, your brain attempts something extraordinary, diving into the deepest, most restorative darkness your biology knows. You'll discover why that dive keeps getting interrupted and understand what your sleeping mind has been quietly asking for all along. Welcome to Sleepy Science Facts. Wherever you are right now, perhaps already settled beneath your blanket, your breathing already a little slower than it was an hour ago. Allow yourself to sink into this particular stillness. We'll journey through 100 sleep science facts about deep sleep, from the brain waves that carry you into the deepest rest, to the obstacles that pull you back too soon, to the quiet science of what your body needs to finally stay there. These calm explorations of the sleeping mind build a quiet community of people learning to rest more deeply. A gentle subscribe, like, or sharing this video creates a pathway for other sleep seekers to find their way here too. One quiet night, one sleepy soul at a time. Sink into your favorite resting spot now. Let your shoulders drop, your jaw soften, your breath grow longer and quieter. You don't need to hold on to any of this sleep science. Just feel that warm, drowsy heaviness beginning to settle through you, like your body is slowly remembering exactly what it was made to do. Close your eyes and breathe deeply. You're resting where deep sleep holds its deepest healing. Let your mind dissolve into that darkness. And now we begin. Deep sleep is one of the most powerful biological states your body ever enters. It is the third stage of the sleep cycle, classified as slow-wave sleep, and named for the long, rolling brain waves that define it. These waves, called delta waves, move at between half a cycle and 4 cycles per second, far slower than the rapid activity your brain produces during waking. During this phase, your muscles are deeply relaxed, your heart rate slows, your blood pressure drops, and your body diverts enormous resources toward repair. You are, in a very real biological sense, doing some of your most important work without doing anything at all. And here is why that matters for tonight. The mystery this science has spent decades untangling is why so many people never reach this state, or reach it only briefly before something pulls them back toward the surface. That is the question we are following through every fact ahead. Your sleep is organized into recurring cycles, each lasting roughly 90 minutes. Within every cycle, you pass through several distinct stages: light sleep, deeper sleep, slow-wave deep sleep, and then REM sleep, the phase associated with vivid dreaming. A typical night of 7 to 9 hours will carry you through 4 to 6 of these cycles. But here is what matters for deep sleep. It is front-loaded. Your first 2 cycles of the night contain the majority of your slow-wave sleep. If you lose those early hours through a late bedtime, alcohol, stress, or interrupted sleep, you lose a disproportionate amount of your deepest rest. The body does not simply reschedule deep sleep for later in the night. It moves on, and the window closes. The brain waves of deep sleep are called delta waves, and they are unlike anything your brain produces while awake. During waking consciousness, your brain generates beta and gamma waves. Fast, small, irregular patterns reflecting active thought and processing. In deep sleep, those busy patterns give way to slow, synchronized sweeps across large areas of the brain. Researchers have described these waves as something like a tide washing across the cortex, a rhythmic, unhurried motion. Each full wave represents millions of neurons firing, and then going quiet in coordinated sequence. This synchrony is essential for the restorative work of sleep. When it is disrupted by noise, stress hormones, or age-related changes, the body feels the difference the next morning in ways that go far beyond simple fatigue. Understanding what disrupts this synchrony is precisely where the science becomes most useful. One of the most transformative discoveries in sleep science came in 2013, when a research team at the University of Rochester published findings that reshaped how scientists understand the sleeping brain. They found that during deep sleep, the brain's glymphatic system, a waste clearance network that uses cerebrospinal fluid, becomes dramatically more active. Slow, rhythmic pulsations of blood vessels during deep sleep appear to pump cerebrospinal fluid through surrounding tissue, sweeping away metabolic waste products. Among those waste products is amyloid beta, the protein fragment that accumulates in the brains of people with Alzheimer's disease. Human evidence for this process has grown substantially since then. A 2025 clinical trial tracking cerebrospinal fluid dynamics confirmed that sleep-active physiological processes enhance overnight clearance of Alzheimer's-associated proteins into the bloodstream, an effect not seen during sleep deprivation. Research is ongoing, but the weight of evidence now points toward deep sleep as a genuinely active clearance process in the human brain. When that deep sleep is cut short, night after night, this clearance may be among the first things lost. Most adults need between 7 and 9 hours of sleep to obtain adequate deep sleep, according to current guidelines from the American Academy of Sleep Medicine. But the amount of slow-wave sleep within that total has its own requirements. Young adults typically spend around 13 to 20% of their total sleep time in slow-wave sleep, roughly 60 to 90 minutes across a full night of 7 to 8 hours. That proportion tends to be highest in the first 2 sleep cycles and decreases steadily with age. It is also not fixed across the lifespan. People who routinely sleep fewer than 6 hours per night show measurable reductions in slow-wave sleep over time, along with associated changes in cognitive function, immune response, and metabolic health. The body can tolerate occasional short nights. It is the chronic shortfall that accumulates, and the deep sleep it costs is not simply reclaimed the following night. The hormone most closely associated with deep sleep is growth hormone. In adults, the majority of the body's daily growth hormone release does not happen during the day. It happens in a concentrated pulse during the first episode of slow-wave sleep, typically within the first hour or 2 of the night. This pulse triggers cellular repair throughout the body. Muscle tissue is rebuilt. Bones are maintained. Fat is metabolized. And the immune system receives support for its overnight operations. Growth hormone levels naturally decline with age, which is part of why the deep sleep of a 60-year-old looks different from the deep sleep of a 20-year-old. But sleep timing and quality also influence how much growth hormone is released. And this pulse is among the first things diminished when deep sleep is repeatedly cut short. Whether by late bedtimes, alcohol, or fragmented sleep architecture, your body temperature plays a quiet but essential role in reaching deep sleep and in staying there in the hours before your natural sleep window. Your core body temperature begins to drop. This cooling is not incidental. It is a biological signal that helps initiate the cascade of changes leading to sleep. The drop in core temperature is accompanied by a redistribution of heat. Blood flows toward your hands and feet, releasing warmth at the extremities. This is why warm hands and feet are actually associated with falling asleep faster. The ideal sleeping environment for most adults has a room temperature somewhere between 60 and 67 degrees Fahrenheit, cool enough to support the body's own temperature-lowering process. Environments that are too warm tend to reduce slow-wave sleep in particular. As the body struggles to achieve the core temperature drop that deep sleep requires, temperature is one of the most direct and manageable factors pulling people out of deep sleep before their biology is finished with it. Adenosine is a chemical compound that builds up in the brain over the course of the waking day. The longer you stay awake, the more adenosine accumulates, and the stronger the drive towards sleep becomes. This accumulated pressure is known as sleep homeostasis, or sleep drive, and it is one of the two main forces regulating your sleep. Deep sleep is the biological mechanism that clears adenosine from the brain. During slow-wave sleep, adenosine is metabolized and removed, and the sleep drive resets toward morning. This is why a night of poor, deep sleep, regardless of total sleep time, often leaves you feeling unrested. The adenosine has not been adequately cleared, and it is also why caffeine works the way it does. Caffeine does not give you energy. It blocks adenosine receptors temporarily, masking the feeling of sleepiness without clearing its underlying cause. The debt remains, and the disruption to deep sleep that follows is part of what makes that debt so difficult to repay. Sleep is regulated by two interacting systems: the homeostatic system, which tracks how long you have been awake and builds sleep pressure accordingly, and the circadian system, which coordinates nearly every biological function in the body to a roughly 24-hour cycle. Deep sleep is most available when these two systems are aligned. When your sleep pressure is high and your circadian clock is signaling that it is nighttime, when they fall out of sync through shift work, jet lag, irregular sleep timing, or simply staying up past your natural sleep window, the coordination breaks down, and deep sleep becomes harder to reach and easier to fragment. Recent research has emphasized that the timing of sleep may be nearly as important as its duration for achieving adequate slow-wave sleep. Misalignment between these two systems is one of the most common and least recognized reasons people cannot stay in deep sleep long enough. The part of the brain most responsible for generating the slow waves of deep sleep is the cerebral cortex, the large, folded outer layer responsible for thought, perception, and memory. During waking, the cortex is in a constant state of activity, receiving and processing information from the senses and generating responses. In deep sleep, the cortex shifts into a very different mode. Large populations of neurons fire together in a slow, sweeping rhythm, then fall temporarily silent, then fire again. This alternation of activity and silence is what produces the characteristic slow oscillations of deep sleep. When these oscillations are disrupted by noise, stress hormones, medications, or age-related changes in the circuits that generate them, deep sleep becomes shallower and more fragmented. So yes, the cortex is where deep sleep lives, and it is also where, most often, it is interrupted. Memory consolidation during deep sleep is not a passive process. While you rest in slow-wave sleep, your hippocampus, a structure deep in the brain, critical for forming new memories, appears to replay the day's experiences in compressed form, transferring information to the cortex for long-term storage. This replay happens at a much faster pace than the original experience and is tightly coupled to the slow oscillations of deep sleep. The timing matters. Slow oscillations from the cortex, sleep spindles generated by the thalamus, and sharp wave ripples from the hippocampus appear to occur in coordinated sequences during slow-wave sleep. Disruptions to any part of this coordination, including disruptions to deep sleep itself, have been shown to impair the consolidation of declarative memories, the kind that involve facts, events, and conscious recall. What interrupts deep sleep does not only interrupt rest, it interrupts the nightly filing of what was learned. Sleep spindles are brief bursts of brain activity that occur during the transitions around deep sleep. Each spindle lasts about half a second to 3 seconds and appears on a brainwave recording as a brief, rapid waxing and waning pattern. They are generated by the thalamus, a structure that acts as a relay station for sensory information, and are thought to serve as a kind of sensory gating mechanism. During deep sleep, spindles help shield the brain from external disturbances, allowing slow-wave activity to continue undisturbed. People with higher spindle density tend to show better memory consolidation and are more able to sleep through noise without waking. Sleep spindle density declines with age, which may be one reason why older adults are more easily pulled out of deep sleep by sounds that would not have disturbed them 20 years earlier. The immune system uses deep sleep for some of its most critical work. During slow-wave sleep, the body releases cytokines, signaling proteins that regulate immune function and help coordinate the response to infection and inflammation. Levels of certain immune cells, including T cells that identify and destroy infected or abnormal cells, follow a daily rhythm that peaks during sleep. Studies examining people who received vaccines found that those who slept adequately after vaccination mounted stronger antibody responses than those who were sleep-deprived. Conversely, even one night of severely disrupted sleep can measurably alter immune cell counts and cytokine profiles in ways that persist for days. The relationship between deep sleep and immune health is now one of the most robustly supported findings in sleep medicine. And it is a relationship that depends not just on sleeping, but on sleeping deeply enough. Your heart and cardiovascular system undergo their own form of restoration during deep sleep. Heart rate slows. Blood pressure drops by 10 to 20%. A phenomenon researchers call nocturnal dipping, and the workload on the heart decreases substantially. This nightly pressure reduction appears to be important for long-term cardiovascular health. People who do not show adequate nocturnal dipping, whether due to poor sleep quality, sleep apnea, or disrupted sleep architecture, show higher rates of hypertension, cardiovascular events, and stroke over time. The drop in blood pressure during deep sleep is not simply a consequence of inactivity. It is an actively regulated process involving the autonomic nervous system, which shifts toward parasympathetic dominance during slow-wave sleep. When deep sleep is cut short, this cardiovascular recovery is cut short with it. And over years, that accumulated deficit leaves its mark on the arteries. Glucose metabolism and insulin sensitivity are both influenced by sleep quality, and deep sleep plays a central role. During slow-wave sleep, the brain's glucose consumption decreases, and the body shifts toward anabolic processes, building and repairing rather than burning. Research has shown that even partial sleep deprivation, reducing sleep to 6 hours or fewer for several consecutive nights, leads to measurable reductions in insulin sensitivity. A landmark study published in the Proceedings of the National Academy of Sciences found that selectively suppressing slow-wave sleep for 3 nights without reducing total sleep time was enough to reduce insulin sensitivity by approximately 25%, a reduction comparable, the researchers noted, to that caused by gaining 20 to 30 pounds. The implication is that it is not just how long you sleep, but how deeply, that matters for metabolic health. Depth, not only duration, is what the body is asking for. The prefrontal cortex, the part of the brain most associated with rational thought, decision-making, and emotional regulation, is one of the brain regions most sensitive to sleep deprivation. After a night of poor deep sleep, activity in the prefrontal cortex tends to decrease, while activity in the amygdala, the brain's emotional alarm center, tends to increase. This shift in balance is associated with greater emotional reactivity, reduced impulse control, and a diminished ability to regulate responses to stress. Researchers studying this dynamic described it as a weakening of the brain's ability to buffer emotional responses, a quieting of the circuits that normally soften reactions to difficult experiences. Adequate deep sleep appears to reset this balance, allowing the prefrontal cortex to reassert its regulatory role over emotional responses the following day. Pain sensitivity changes measurably with sleep quality. Studies using standardized pain threshold tests have consistently found that sleep-deprived individuals, and particularly those lacking adequate deep sleep, report lower pain thresholds and greater sensitivity to discomfort. The mechanisms involve multiple pathways, changes in the activity of descending pain-modulating systems, alterations in opioid receptor sensitivity, and shifts in inflammatory markers. Chronic pain and poor sleep tend to reinforce each other in a cycle that can be difficult to interrupt. People with conditions such as fibromyalgia, arthritis, or lower back pain frequently show disrupted sleep architecture with reduced slow-wave sleep. And interventions that improve sleep quality often produce measurable reductions in reported pain, even without any change in the underlying physical condition. The pain had not changed. The depth of rest had, and that, it turned out, was enough. The skin renews itself most actively during sleep. Human growth hormone, released in the first deep sleep episode of the night, stimulates cell division and protein synthesis throughout the body, including in the skin's basal layer, where new cells are constantly being generated to replace those lost at the surface. Collagen synthesis, which gives skin its elasticity and firmness, also follows a circadian rhythm that peaks during sleep. Research has connected chronic sleep insufficiency to increased skin aging, reduced barrier function, and slower recovery from ultraviolet exposure. The phrase beauty sleep reflects something genuinely biological. The skin is, among other things, a tissue that does some of its most productive work in the dark, and one that registers, quite visibly. When deep sleep has been repeatedly denied, children spend far more time in deep sleep than adults. Infants may spend up to 50% of their sleep time in slow-wave sleep, while school-age children typically spend 25 to 30%. This proportion decreases steadily through adolescence and into adulthood. The high proportion of deep sleep in children is thought to reflect the extraordinary demands of brain development during the early years. Slow-wave sleep supports synaptic pruning, the process by which the brain refines its connections, removing synapses that are less used and strengthening those that are more active. This pruning is essential for efficient neural function. The fact that deep sleep decreases as development slows suggests that the brain is calibrating its restorative mechanisms to match its current needs. and beginning that recalibration far earlier in life than most people realize. One of the most underappreciated obstacles to deep sleep is light exposure in the evening hours. The human eye contains specialized cells called intrinsically photosensitive retinal ganglion cells, which contain a photopigment called melanopsin. These cells are particularly sensitive to short-wavelength blue light, the kind emitted abundantly by LED screens and energy-efficient lighting. When these cells detect blue light in the evening, they send signals to the brain's master circadian clock, which in turn suppresses the release of melatonin. Melatonin does not directly cause deep sleep, but it facilitates the shift in internal timing that makes deep sleep possible. Suppressing it with evening light delays the entire sleep architecture, often compressing the time available for slow-wave sleep. This is among the most modifiable reasons people cannot stay in deep sleep long enough, and one of the simplest to begin addressing. Alcohol is one of the most widely misunderstood substances in relation to sleep. Many people use it to fall asleep more easily, and it does indeed reduce the time it takes to fall asleep. But alcohol disrupts sleep architecture in ways that become apparent later in the night. It suppresses REM sleep in the first half of the night and fragments slow-wave sleep in the second half. As the liver metabolizes it and blood alcohol levels fall, the result is a night that may begin with rapid sleep onset but progresses into lighter, more fragmented sleep than would occur without alcohol. People who drink regularly and then stop often experience a rebound of vivid, disturbing dreaming as REM sleep returns, a sign of how profoundly alcohol had been suppressing it. For deep sleep specifically, alcohol is not a sedative. It is a disruptor that wears the appearance of one. The thalamus, a paired structure deep in the brain, acts as a kind of gatekeeper for sensory information during sleep. During waking, it relays incoming signals from the senses to the cortex for processing. During deep sleep, it shifts into a different mode, generating rhythmic bursts of activity that help synchronize cortical slow waves and simultaneously reduce the transmission of incoming sensory signals to the cortex. This gating function helps explain why we can sleep through moderate background noise but are awakened by something personally significant. A baby's cry, a partner's unusual breathing, or our own name spoken softly. The thalamus is not simply blocking all signals, it is selectively filtering them, remaining alert to what matters while allowing irrelevant stimuli to pass without waking us. When this gating mechanism is compromised by stress, by certain medications, by age, the filtering becomes leaky, and deep sleep becomes harder to hold. Recent research has brought increasing attention to the role of the gut-brain axis in sleep quality. The gut microbiome, the community of trillions of microorganisms living in the digestive tract, communicates with the brain through multiple pathways, including the vagus nerve and the enteric nervous system. The amino acid tryptophan, absorbed from food, serves as the biological precursor for both serotonin and melatonin, with melatonin ultimately synthesized in the pineal gland from that upstream chain, while gut serotonin and brain serotonin remain in separate pools that do not cross the blood-brain barrier. The availability of tryptophan across the gut-brain axis may be one pathway through which microbiome health influences sleep quality. Studies in both animal models and human populations have found associations between microbiome diversity and sleep quality, with more diverse microbiomes generally associated with better sleep metrics. The mechanisms are still being clarified, but the connection adds a dimension to sleep health that goes well beyond the brain alone. Exercise is one of the most reliably effective interventions for improving deep sleep. Aerobic exercise, in particular. Walking, running, cycling, swimming, has been shown to increase the proportion of slow-wave sleep and to improve its depth. The mechanisms are multiple. Exercise increases adenosine production, which builds sleep pressure. It raises core body temperature, which then falls during recovery, initiating sleep-promoting signals, and it reduces anxiety and stress, which are among the most common disruptors of deep sleep. Resistance training also shows positive effects on slow-wave sleep, though the research is less extensive. The timing of exercise matters somewhat. Vigorous exercise very close to bedtime may delay sleep onset for some people, but the overall effect of regular physical activity on sleep depth is robustly positive. For many people, this is one of the most direct levers available for recovering the deep sleep that lifestyle has been gradually reducing. Stress and the hormone cortisol are among the most potent suppressors of deep sleep. Cortisol follows a circadian rhythm. It is highest in the morning, supporting alertness and activity, and lowest in the early part of the night, allowing the deeper stages of sleep to emerge. When the stress response system is chronically activated, cortisol levels remain elevated in the evening and overnight, disrupting the hormonal conditions that slow-wave sleep requires. Psychological stress, financial worry, relationship tension, and unresolved conflict have all been shown to increase overnight cortisol levels and reduce slow-wave sleep percentage. This creates a self-reinforcing cycle. Stress disrupts deep sleep, and insufficient deep sleep impairs the brain's ability to process and regulate emotional responses to stress the following day. Of all the forces working against sustained deep sleep, chronically elevated cortisol may be the most pervasive and the most invisible to the person carrying it. Let's pause here and notice your body. As you breathe out slowly, appreciate each tiny cell inside, trillions of organisms working in silence, heart beating, lungs breathing, brain thinking, all without your command. Right now, while you rest here in this quiet, your brain is doing exactly what it was built to do, sorting the day, clearing what is no longer needed, filing what matters, releasing what does not. You don't need to do anything. You only need to be here. This is the oldest thing a human body knows how to do. Now, we continue. Sleep architecture. The pattern of sleep stages across the night is not fixed. It is shaped by dozens of variables. Your age, your health, your stress levels, your sleep timing, what you ate, how much light you are exposed to, and even the season. Researchers who track sleep architecture describe it as more like a dynamic landscape than a fixed schedule. The body is always attempting to reach deep sleep and always navigating around whatever obstacles stand in its way. Understanding why deep sleep is so difficult to sustain is not a matter of finding one cause. It is a matter of understanding an entire ecology, a set of interacting conditions that either support or undermine the body's nightly attempt to restore itself. Most people are contending with several of these conditions simultaneously, without knowing it. Sleep apnea is one of the most common medical conditions disrupting deep sleep globally. It occurs when the airway partially or fully collapses during sleep, causing breathing to stop briefly, sometimes hundreds of times per night. Each cessation triggers a brief arousal as the brain detects the drop in oxygen and signals the airway muscles to reopen. These arousals are often too brief to be remembered upon waking, but they are sufficient to fragment sleep architecture and prevent the sustained slow-wave activity that characterizes deep sleep. Estimates suggest that approximately 1 billion people worldwide have some degree of obstructive sleep apnea, and a substantial proportion remain undiagnosed. The condition is associated with cardiovascular disease, metabolic syndrome, cognitive decline, and depression, many of which may be mediated, at least in part, by the chronic deprivation of deep sleep it quietly causes. Night after night, aging is one of the most universal factors reducing deep sleep. The amount of slow-wave sleep a person obtains begins declining after adolescence, with measurable reductions continuing steadily through adulthood. By the time most people reach their 60s, slow-wave sleep may represent only 5 to 8% of total sleep time, compared to 15 to 20% in the early adult years. The mechanisms include age-related changes in the slow oscillation-generating circuits of the cortex, reductions in growth hormone levels, changes in circadian rhythm, timing, increased prevalence of sleep-disrupting conditions such as sleep apnea and restless leg syndrome, and altered sensitivity to adenosine. Some of this decline is considered a normal feature of aging, but research increasingly suggests it is not entirely inevitable. Lifestyle, health management, and sleep timing practices can meaningfully influence how much slow-wave sleep is preserved across the decades. Caffeine has a half-life in the human body of approximately 5 to 7 hours, meaning that half of the caffeine from a cup of coffee consumed at noon is still present in the bloodstream at 6 or 7 in the evening. For most people, this residual caffeine does not prevent them from falling asleep, but it does alter the architecture of the sleep they obtain. Research using polysomnography has shown that caffeine consumed even 6 hours before bedtime measurably reduces slow-wave sleep, even in people who feel they have slept normally. The mechanism is adenosine blockade. By occupying adenosine receptors, caffeine reduces the sleep pressure signal that helps drive the brain into deep sleep. Habitual caffeine consumers may be partially tolerant to this effect, but tolerance is never complete. The disruption to slow-wave sleep persists at some level, and for many people, it is a daily, invisible reason they cannot stay in deep sleep as long as their body needs. The bedroom environment has measurable effects on sleep architecture. Noise is particularly disruptive to deep sleep because the brain remains partially responsive to sound even during slow-wave sleep, and sudden or unpredictable sounds trigger microarousals that can shift the sleeper out of deep sleep without fully waking them. Research conducted in hospital settings has documented substantial reductions in slow-wave sleep among patients due to overnight noise, with implications for recovery rates and cognitive function. Continuous background sounds, such as white noise, pink noise, or recordings of nature, have shown some ability to mask disruptive sounds and may help stabilize sleep architecture. Pink noise, which has more energy in lower frequencies than white noise, has received particular research attention after studies found it could enhance slow oscillation power during sleep. The environment we sleep in is not neutral. It either supports deep sleep or works against it, moment by moment through the night. The suprachiasmatic nucleus, a tiny cluster of approximately 20,000 neurons in the hypothalamus, is the master clock of the human body. It receives light information directly from the retina and uses it to synchronize the body's circadian rhythms to the external day-night cycle. It then sends timing signals throughout the brain and body, coordinating the release of hormones, the regulation of body temperature, the activity of the immune system, and the architecture of sleep, including when slow-wave sleep is most available. When the suprachiasmatic nucleus is well entrained to the light-dark cycle, deep sleep occurs at predictable, biologically optimal times. When it is misaligned, through irregular schedules, artificial light exposure, or travel across time zones, the timing of deep sleep becomes unpredictable and less accessible. The clock does not stop, but it can be shifted. And when it is, deep sleep pays the cost. Melatonin is often described as the sleep hormone, but its role is more accurately that of a darkness signal. The pineal gland begins releasing melatonin in the evening as light levels drop, signaling to the body that night has arrived. Melatonin does not directly induce sleep. Rather, it coordinates the biological timing that makes sleep, and specifically deep sleep, available at the appropriate time. Supplemental melatonin at low doses, taken at the right time relative to the internal clock, can help shift sleep timing in people who are misaligned, such as those with delayed sleep phase syndrome or jet lag. But high doses of supplemental melatonin do not necessarily produce deeper sleep, and some research suggests they may alter sleep architecture in complex ways. The body's own melatonin, released in small amounts at precisely the right time, remains the most physiologically appropriate version, and the one most easily disrupted by evening light. The brainstem contains several groups of neurons that are essential for sleep regulation. In the pons and medulla, circuits control the transitions between wakefulness, REM sleep, and non-REM sleep. During deep sleep, neurons in the ventrolateral preoptic area of the hypothalamus become highly active, releasing GABA, the brain's primary inhibitory neurotransmitter, and inhibiting the arousal-promoting circuits of the brainstem and basal forebrain. This inhibition is what allows the cortex to enter the slow-wave state. When the ventrolateral preoptic area is suppressed by certain medications, by injury, or by the natural changes of aging, the ability to maintain deep sleep is correspondingly reduced. The circuits that generate deep sleep are themselves vulnerable to the conditions of the life lived around them. Restless leg syndrome affects an estimated 5 to 10% of adults in the United States and is one of the more common neurological conditions, specifically disrupting deep sleep. It is characterized by an uncomfortable urge to move the legs, which worsens at rest and in the evening, precisely when the body is attempting to transition into sleep. The neurological basis involves disruptions in the dopaminergic system, particularly in pathways involving iron metabolism in the brain. Iron deficiency, even without anemia, is associated with more severe symptoms. Periodic limb movement disorder, a related condition involving repetitive limb movements during sleep, occurs in a high proportion of people with restless leg syndrome and produces microarousals that fragment sleep architecture, repeatedly pulling the sleeper out of the slow-wave episodes that would otherwise develop across the early hours of the night. The emotional content of the day influences the structure of the following night's sleep. Research using mood assessments and polysomnography has found that days characterized by negative emotional states, anxiety, anger, sadness, or rumination, are associated with increased sleep fragmentation and reduced slow-wave sleep the following night. Positive emotional states and experiences of social connection. By contrast, tend to be associated with more consolidated sleep architecture. The mechanism involves the stress response system. Negative emotional arousal activates it, elevating cortisol and other stress hormones that persist into the night. This is one reason why cognitive behavioral therapy for insomnia, which addresses the thought patterns and emotional responses associated with sleep, has shown effects on sleep architecture, not just on sleep onset and duration. The emotional residue of the day is among the quietest and most persistent disruptors of the night. The foods you eat can influence how deeply you sleep through several interacting mechanisms. High glycemic index meals consumed close to bedtime have been associated with more fragmented sleep in some studies, possibly through effects on blood sugar fluctuations overnight. A diet rich in fiber has been associated with more time in deep sleep and less time in lighter sleep stages. According to research published in the Journal of Clinical Sleep Medicine, the amino acid tryptophan, found in turkey, dairy, nuts, and seeds, is a precursor to both serotonin and melatonin through a pathway that ultimately synthesizes melatonin in the pineal gland. And its availability in the diet is one factor influencing the timing and depth of sleep. Magnesium, found in leafy greens, legumes, and nuts, appears to support slow-wave sleep through its effects on the GABA system. The interaction between diet and sleep architecture is nuanced, but the choices made at the dinner table cast a longer shadow into the night than most people realize. Body position during sleep influences breathing, and breathing quality is one of the primary determinants of sleep architecture depth. Sleeping on the back, the supine position, increases the likelihood of airway obstruction and snoring in people with any degree of anatomical narrowing of the airway. The tongue and soft tissues of the throat are more likely to fall backward under gravity in this position, partially obstructing the airway and increasing the work of breathing. Side sleeping generally produces more open airways and is recommended for people with sleep apnea or a tendency towards snoring. The connections between sleep position and sleep architecture extend to deep sleep specifically. In people with airway narrowing, the supine position produces more microarousals during slow-wave episodes, interrupting the very rest the body was working hardest to achieve. Napping has a complex relationship with nighttime deep sleep. A nap of 20 to 30 minutes, often called a power nap, reduces adenosine levels modestly and can improve alertness and performance without significantly depleting the sleep pressure needed for nighttime deep sleep. Longer naps of 60 to 90 minutes, which allow entry into slow-wave sleep, provide more substantial restoration but can noticeably reduce sleep pressure for the following night, making it harder to achieve deep sleep at the appropriate time. The timing of the nap matters as well. Naps taken in the early afternoon, aligned with a natural post-lunch dip in alertness, tend to have less impact on nighttime sleep architecture than naps taken in the late afternoon or evening. For people already struggling to maintain deep sleep at night, daytime napping is one of the factors most worth examining carefully. The relationship between mental health and deep sleep is bidirectional and deeply interconnected. Depression is associated with profound changes in sleep architecture, including reduced slow-wave sleep, shortened REM sleep latency, and early morning awakening. Anxiety disorders are associated with hyperarousal of the central nervous system, which interferes with the cortical synchrony required for slow-wave sleep. Post-traumatic stress disorder involves some of the most severely disrupted sleep architectures documented in clinical research. With fragmented sleep, nightmare prevalence, and marked reductions in slow-wave sleep. The connections run in both directions. Sleep deprivation experiments reliably produce symptoms resembling depression and anxiety in healthy participants, and treatments that improve sleep quality, including both pharmacological and behavioral interventions, often produce measurable improvements in mental health outcomes. Deep sleep does not just reflect mental health, it actively maintains it. Seasonal changes affect sleep architecture in ways that are only beginning to be fully documented. The reduced daylight of autumn and winter months delays melatonin onset for some people, compressing the time window available for deep sleep. A large-scale study published in 2023 in the journal Current Biology, which analyzed sleep data from participants across multiple seasons, found that total sleep time was longer in winter and that sleep architecture showed seasonal variation, with some participants showing altered proportions of deep and REM sleep across the year. These changes are consistent with the hypothesis that humans retain vestiges of seasonal sleep regulation, even in modern environments with artificial lighting. Seasonal affective disorder, which involves depressed mood and altered sleep patterns during winter months, may represent one end of a continuum of biological responses to seasonal light changes, and one more layer in the complex ecology of what prevents deep sleep from lasting as long as the body needs. You're still here, still listening, still somewhere between thinking and resting. Take one slow breath. Your brain is doing exactly what it was built to do tonight. We're moving through the science of deep sleep together. You and I, and every quiet mind listening in the dark. No rush, no destination, just the gentle current of curiosity carrying us forward. Let's continue. Temperature regulation during sleep is not simply a matter of comfort. The body actively manages heat redistribution throughout the night, and disruptions to this process, whether from an overly warm environment, fever, or hormonal changes, directly impair sleep architecture. During the transitions into deep sleep, cutaneous blood flow increases significantly, redistributing heat from the body's core to its periphery. This process is regulated by the hypothalamus, which acts as the body's thermostat. Research has found that warming the skin artificially, through warm baths before bed or wearing warming socks, can accelerate the heat redistribution process and reduce the time it takes to reach deep sleep. The underlying signal to the brain is consistent. When heat is leaving the body efficiently, the conditions for slow-wave sleep are being met. When it is not, those conditions remain just out of reach. The brain, during deep sleep, is not inactive. It is working differently. Neuroimaging studies have shown that while overall metabolic activity in the cortex decreases during slow-wave sleep, specific regions remain active in coordinated ways. The thalamus and brainstem maintain activity that supports the generation of sleep rhythms. The hippocampus continues its memory consolidation operations, and regions involved in emotional processing show patterns of activity that are distinct from both waking and REM sleep. The picture that has emerged from decades of neuroimaging research is one of a brain using the reduced sensory demands of sleep to attend to its own internal housekeeping, consolidating, repairing, clearing, and organizing in ways that waking activity does not allow. When this housekeeping is interrupted by fragmented deep sleep, those tasks are left unfinished, and the brain carries that incompleteness into the following day. The electroencephalographic signature of deep sleep, the slow delta waves, can be enhanced or suppressed by a range of pharmaceutical agents, benzodiazepine medications, commonly used for anxiety and sleep disorders, suppress slow-wave sleep while increasing lighter sleep stages, which is one reason why people who take them often report sleeping longer but not feeling more rested. Certain antidepressants, including SSRIs and SNRIs, also reduce slow-wave sleep in many people. Conversely, some newer sleep medications designed to target specific receptors, including drugs that block the neuropeptide orexin, which promotes wakefulness, appear to produce sleep with a more natural architecture, including preserved or enhanced slow-wave sleep. The pharmacology of deep sleep is an active area of research, and the field is gradually moving away from sedation models toward architecture-preserving approaches. Sound during sleep has effects that go beyond simple wakefulness. Researchers have explored the possibility of using sound to enhance slow-wave sleep, a concept sometimes called acoustic slow-wave sleep enhancement. By playing brief bursts of pink noise or other sounds timed to coincide with the upward phase of slow cortical oscillations, some studies have been able to increase the amplitude of slow waves and the amount of time spent in deep sleep. The effect appears to rely on a resonance between the external auditory stimulus and the brain's own oscillatory rhythm. Early findings from several European research groups suggested improvements in memory consolidation following acoustically enhanced sleep. The technique has not yet been validated for broad clinical use, but it illustrates the degree to which the sleeping brain remains receptive to carefully timed external signals, and suggests that the conditions supporting deep sleep can be gently cultivated. Not only removed of obstacles, physical pain is one of the most common reasons people fail to maintain deep sleep. Both acute and chronic pain activate the same arousal circuits that respond to stress and threat, making it biologically difficult for the brain to sustain the cortical synchrony required for slow-wave sleep. Research has found that people with chronic low back pain show electroencephalographic patterns during slow-wave sleep that are interspersed with alpha-wave intrusions, the brain waves associated with relaxed wakefulness, creating a state sometimes described as alpha-delta sleep. This pattern is associated with non-restorative sleep and is found in people with fibromyalgia, rheumatoid arthritis, and other chronic pain conditions. Treating the underlying pain condition consistently improves sleep architecture, and improving sleep architecture through other means often reduces the perceived intensity of chronic pain, suggesting a genuine bidirectional relationship in which each holds the other in place. The relationship between deep sleep and learning is well established at the cellular level, a process called long-term potentiation, the strengthening of synaptic connections through repeated activation, is believed to underlie the formation of long-term memories. Research supports what is known as synaptic homeostasis, the idea that slow-wave sleep involves the selective weakening of synapses that were broadly strengthened during the waking day, preserving the meaningful patterns while reducing metabolic demand. This theory, developed by researchers Giulio Tononi and Chiara Cirelli, proposes that slow-wave sleep is essential for keeping the brain efficient and adaptable, not just storing information, but maintaining the capacity to continue learning. Supporting evidence from human studies has shown that slow-wave sleep following a learning session enhances subsequent recall performance, and disrupting slow-wave sleep specifically impairs that improvement. Every interruption to deep sleep is, in some sense, an interruption to the night's learning. Obesity and sleep architecture interact in several important ways. Excess body weight, particularly central adiposity around the abdomen and neck, increases the likelihood of obstructive sleep apnea and upper airway resistance syndrome, both of which fragment deep sleep, but the relationship extends beyond the mechanical. Adipose tissue, especially visceral fat, is metabolically active and produces inflammatory signaling molecules that have been shown in research to disrupt sleep architecture. A large body of evidence from longitudinal studies has also found that insufficient sleep, and particularly insufficient deep sleep, is associated with weight gain over time through effects on appetite-regulating hormones, ghrelin, which stimulates hunger, increases with sleep deprivation, leptin, which signals satiety, decreases. The steady rise in metabolic disease globally involves multiple interacting factors, and the role of chronic sleep deprivation among them is now well enough established that sleep is increasingly included in metabolic health guidelines. Deep sleep has a protective relationship with cardiovascular risk. A study published in 2023 in the journal JAMA Cardiology, which followed participants over a period of years, found that lower amounts of deep sleep were associated with a higher risk of coronary artery disease. Independent of other cardiovascular risk factors, the mechanisms proposed include the role of deep sleep in blood pressure regulation, inflammation reduction, and sympathetic nervous system quieting during slow-wave sleep. The balance between the sympathetic nervous system, which accelerates heart rate and raises blood pressure, and the parasympathetic nervous system, which slows heart rate and lowers blood pressure, shifts substantially in favor of the parasympathetic. Nights of insufficient deep sleep mean nights of inadequate cardiovascular recovery, and accumulated over years. That exposure may translate into measurable changes in arterial health. More recent research has begun to map the specific routes through which cerebrospinal fluid moves during deep sleep and to identify what drives that movement. A landmark study published in the journal Cell in early 2025 identified norepinephrine-mediated slow pulsations of blood vessels as the primary driver of glymphatic fluid flow during non-REM sleep. These slow oscillations, occurring roughly every 50 seconds, appear to create a pumping mechanism that circulates cerebrospinal fluid through the surrounding brain tissue. Separately, a 2025 clinical trial in humans directly tracked clearance of amyloid beta and tau proteins from the brain into the bloodstream during normal sleep compared to sleep deprivation, finding that sleep-active physiological processes significantly enhance overnight clearance of these Alzheimer's-associated biomarkers. The picture emerging from this new wave of research is not simply that the brain clears waste during sleep. It is that deep sleep is the primary engine of that clearance, and that what cuts it short also cuts that engine short. We are now halfway through our journey, and a picture has been emerging. Fact by quiet fact. Deep sleep is not one thing. It is a state that serves the brain, the body, the immune system, the cardiovascular system, the metabolic system, and the emotional regulation system simultaneously. It is a window, open for perhaps 60 to 90 minutes each night, during which the body performs work it cannot perform any other way. The reason so many people cannot stay in deep sleep long enough is not usually a single cause. It is more often a landscape of small interferences: the wrong timing, the wrong light, the wrong substances, the unresolved worry, The aging body. The misaligned clock. Understanding that landscape is the beginning of learning to navigate it, and the second half of this journey begins to map the way every cell you have today will be completely replaced within 7 years. You are in the middle of a continuous renewal, not the same body that learned to walk, or first tasted something sweet, constantly becoming. Tonight is part of that process. This rest, this quiet, this particular depth of darkness, right now, in the slow drift between wakefulness and sleep. Something is being remade. You don't need to know what. You only need to be here while it happens. And we continue the journey. Chronic sleep restriction. Sleeping less than the body requires. Night after night. Produces a type of cognitive impairment that is particularly deceptive, unlike acute total sleep deprivation, which produces obvious and subjectively recognized impairment. Chronic partial sleep deprivation impairs performance while leaving the person unaware of how impaired they have become. Research by David Dingers and colleagues at the University of Pennsylvania found that participants restricted to 6 hours of sleep per night for 2 weeks showed performance deficits equivalent to 2 nights of total sleep deprivation, yet reported feeling only slightly sleepy. The slow-wave sleep that is lost in chronic restriction is not recovered simply by sleeping normally for 1 or 2 nights. Full recovery of baseline cognitive function appears to require multiple nights of adequate, uninterrupted sleep, and the deficit accumulates faster than most people recognize it doing. The timing of meals interacts with sleep architecture through the body's circadian system. The digestive system follows its own circadian rhythms. Influenced by the timing of food intake, eating large meals close to bedtime increases core body temperature through the thermogenic effect of digestion, directly working against the temperature drop needed for deep sleep. Late eating also delays gastric emptying, which can cause discomfort during sleep, and alters the release of insulin and other metabolic hormones in ways that have been associated with altered sleep architecture. Research has found that time-restricted eating, limiting food intake to a consistent 8 to 10 hour window during the daytime, can improve circadian alignment and has shown preliminary positive effects on sleep quality in some populations. The gap between the last meal and bedtime is not simply a digestive matter, it is a sleep architecture matter. Social isolation and loneliness have measurable effects on sleep architecture. Research found that loneliness was associated with more fragmented sleep, more nighttime awakenings, and reduced slow-wave sleep. The proposed mechanism involves the heightened alertness associated with perceived social isolation. The brain of a lonely person may remain in a state of elevated vigilance, even during sleep, that interferes with the sustained cortical synchrony of deep sleep. This is consistent with evolutionary models suggesting that sleeping alone or in socially isolated conditions would historically have represented a vulnerability requiring heightened awareness. The body does not always update its responses in line with modern circumstances. When social connection is absent during the day, the sleeping brain at night may still be scanning for threat, and that quiet scanning is enough to keep deep sleep from fully settling. Mindfulness meditation has been studied as an intervention for improving sleep quality, and a growing body of research supports its effects on sleep architecture. A meta-analysis published in JAMA Internal Medicine found that mindfulness meditation programs produced significant improvements in insomnia, fatigue, and depression in people with sleep problems. More recent studies using electroencephalography have found that regular mindfulness practice is associated with changes in slow-wave sleep metrics, including increased slow oscillation amplitude in some participants. The mechanisms may involve reduced cortisol secretion, reduced default mode network activity before sleep, and changes in how the mind responds to the hypnagogic experiences. The sensory fragments and thoughts that occur at the threshold of sleep, that often cause people to reawaken. Practices that quiet the mind during the day appear to have genuine, measurable effects on how deeply the mind can rest at night. The effects of deep sleep on physical performance are well documented in sports science. Studies in elite athletes have consistently found that extending sleep duration, and specifically increasing time in slow-wave sleep, produces measurable improvements in reaction time, accuracy, power output, and injury recovery rates. The mechanisms include increased growth hormone release, reduced inflammatory markers, faster muscle glycogen resynthesis, and enhanced motor skill consolidation, which relies on both slow-wave sleep and REM sleep. The National Basketball Association, National Football League, and other professional sports organizations have increasingly incorporated sleep science into their athlete performance programs, recognizing sleep architecture quality as a legitimate performance variable. Sleep extension studies in collegiate basketball players, tennis players, and swimmers have consistently shown that more and better deep sleep translates into measurable athletic advantage. What disrupts deep sleep disrupts recovery in ways that show up on the court and on the track. The depth of deep sleep, measured by the amplitude of slow cortical oscillations, varies across the scalp in patterned ways. Slow waves are not generated uniformly across the cortex. They tend to originate most powerfully over the frontal and prefrontal cortex and propagate backward toward the occipital and parietal regions. Research has found that the regions of the cortex most active during the preceding waking day tend to generate the strongest slow-wave activity during the following night's sleep. This is a distinct but complementary finding to synaptic homeostasis, where the theory speaks to what slow waves do to synapses. This topographic pattern speaks to where they do it most intensely. The circuits that worked hardest during the day received the most concentrated overnight attention. The brain does not treat all of its regions equally during deep sleep. It prioritizes the ones that most need it. The default mode network, a set of brain regions that become active during rest, daydreaming, and self-referential thinking, shows a distinctive pattern of activity during the transition into deep sleep. As slow-wave sleep deepens, default mode network activity decreases, and its internal connectivity breaks down. This dissolution of the default mode network may be part of what allows consciousness to narrow during deep sleep. The inward-focused thinking that characterizes mind-wandering and self-reflection gives way to a more fundamental state. People who have difficulty quieting their default mode network, those prone to rumination, anticipatory worry, or self-critical thought, often have trouble making this transition smoothly and may cycle in and out of the lighter stages of sleep rather than descending into sustained, slow-wave activity. The busy mind and the deep sleeping brain are, neurologically, almost opposite states. Thermoregulation during sleep differs between men and women, and these differences have been proposed as one factor contributing to the higher rates of insomnia reported by women. Women's core body temperature tends to fluctuate more substantially across the menstrual cycle and during the luteal phase, the 2 weeks before menstruation. Elevated progesterone levels raise core temperature slightly, which may make the temperature drop needed for deep sleep harder to achieve. During perimenopause and menopause, vasomotor symptoms, hot flashes and night sweats, are among the most common disruptors of sleep architecture, specifically interrupting deep sleep episodes. Research has found that women in midlife show more fragmented slow-wave sleep than age-matched men, a difference that narrows after the menopausal transition is complete. Hormone therapy has shown benefits for sleep architecture quality in some studies of perimenopausal women, suggesting that for some, the obstacle to sustained deep sleep is hormonal and addressable. The cerebellum, traditionally associated with movement coordination and balance, has emerged in recent research as an active participant in sleep regulation. Studies in rodents and, more recently, in humans have found that the cerebellum shows sleep-related activity patterns distinct from those of the cortex, and that cerebellar circuits communicate with brainstem and thalamic sleep-regulating areas. Some researchers have proposed that the cerebellum may play a role in the motor quiescence of deep sleep, the reduction of muscle tone and movement that allows the body to rest fully. The cerebellum is densely connected to the limbic system through indirect pathways, and its emerging role in emotional processing may also be relevant to the emotional restorative functions of deep sleep. The brain regions involved in deep sleep continue to expand with each year of research, a reminder that the science of sleep is still, in many ways, at its beginning. Light in the red to near-infrared wavelength range has been explored as a potential tool for improving sleep quality. Unlike blue light, which suppresses melatonin through melanopsin-containing retinal cells, long-wavelength red light does not significantly activate melanopsin pathways and therefore does not suppress melatonin in the evening. Some preliminary studies have found that exposure to red or near-infrared light in the evening may be associated with improvements in certain sleep metrics. The mechanism proposed in some animal research involves photobiomodulation of mitochondrial function in brain cells. This remains a field of early inquiry, and the evidence is not yet strong enough to support clinical recommendations. But it illustrates the increasingly sophisticated lens through which researchers are beginning to examine the light-sleep relationship, and the possibility that the quality of light in the hours before sleep matters in ways beyond simple brightness. Dreams occur in all sleep stages, but the nature of dreaming differs between slow-wave sleep and REM sleep. During deep sleep, if dreamlike experiences occur, they tend to be fragmentary, thought-like, or characterized by simple perceptions rather than the vivid narrative scenarios of REM sleep. Some researchers have described slow-wave sleep mentation as being more like an unfinished thought than a story. The brain mechanisms involved are consistent with this. The dorsolateral prefrontal cortex, which is necessary for narrative construction and self-reflective awareness, shows reduced activity during deep sleep. The experience of deep sleep from the inside is often described by people awakened from it as a kind of blankness, not a story, not a landscape, but simply the absence of awareness. This absence is not empty. It is the condition under which the brain does its most essential maintenance. The connection between deep sleep and the gut microbiome has been explored in several recent studies. A study published in PLOS One, analyzing data from a cohort of adults, found associations between specific microbiome composition metrics and sleep quality measures, including sleep efficiency and time in deeper sleep stages. Certain bacterial genera showed correlations with sleep quality in ways that were independent of diet and other confounders. The directionality of causation is not yet established, and the field is acknowledging the complexity of the gut-brain axis with appropriate caution. But the emerging picture suggests that the ecological community of the gut is part of the broader biological context in which sleep, including deep sleep, either flourishes or struggles. Caring for the gut is, in ways, still being mapped. Also caring for the depth of the night. The transition from slow-wave sleep into REM sleep, and then back into lighter non-REM sleep to begin a new cycle, is regulated by competing neurochemical systems, neurons that produce norepinephrine and serotonin, which support arousal and wakefulness, are quietest during deep sleep. Neurons that produce acetylcholine, which supports the active, dreaming brain of REM sleep, gradually increase in activity as a deep sleep episode progresses, eventually tipping the balance and initiating the transition to REM. This cycling between deep sleep and REM is not simply a neutral alternation. The body appears to use REM sleep to process the emotional material that was not fully integrated during the preceding day, working on the texture and meaning of experience while the cortex is less analytically engaged. The two states are complementary. Neither replaces the other. And when deep sleep is cut short, REM loses part of the foundation it builds upon. The phenomenon of sleep inertia, the grogginess and disorientation felt upon waking, is strongest after being awakened from deep sleep. The brain needs time to shift from the low-frequency, high-amplitude activity of slow-wave sleep to the faster rhythms of wakefulness. And during this transition, cognitive performance and reaction time are significantly impaired. Sleep inertia is worst when the awakening is timed against the body's natural sleep pressure, such as waking from a midday nap that entered slow-wave sleep, or being woken in the early part of the night when slow-wave pressure is highest. Its presence is, in a sense, confirmation that the body was doing something important. The grogginess of true deep sleep awakening is the biological equivalent of being interrupted mid-sentence. The feeling of a task left undone. We have now learned enough to understand the answer the science has been building toward. The reason you cannot stay in deep sleep long enough is almost never a failure of your body's intention. The body always tries. It builds the pressure. It drops the temperature. It releases the hormones. It quiets the arousal systems. It reaches for the slow oscillations that define this most restorative state. What interrupts it, again and again, is the environment it is trying to do this work within. The noise, the light, the residual caffeine, the elevated cortisol, the structural obstruction in the airway, the mistimed schedule, the accumulated weight of unprocessed emotion. The body is not broken. It is attempting something exquisitely calibrated, in a world that was not designed with that calibration in mind. The answer to the question is not a diagnosis. It is a map. Sleep education is increasingly recognized as a public health issue. Surveys conducted in the United States consistently find that a substantial minority of adults report sleeping fewer than 7 hours per night. Data from the Centers for Disease Control and Prevention has identified insufficient sleep as a public health concern, linked to motor vehicle accidents, occupational errors, chronic disease burden, and mental health costs. Despite this recognition, sleep is still widely treated in modern culture as a variable, something to be optimized away from. rather than a biological necessity equivalent in importance to food and water. This cultural framing has real consequences for the depth and quality of sleep that millions of people actually obtain. Changing it begins with understanding what is being lost. And that is precisely what this journey has been about. The relationship between screen use and sleep architecture has been documented across multiple studies and age groups, beyond the direct effect of blue light suppressing melatonin. Screen use in the hour before bed is associated with cognitive and emotional arousal, the opposite of the conditions that support slow-wave sleep onset. Stimulating content, social media, news, competitive games, activates the dopaminergic reward system and the amygdala's emotional processing circuits, maintaining the brain in a state of wakefulness that is difficult to quickly reverse, even after putting down the screen. The neural state induced by engaging content persists for some time. Research with adolescents has found particularly strong associations between evening screen use and reduced slow-wave sleep, in part because the adolescent brain is already biologically shifted toward later sleep timing and is therefore more vulnerable to delays in sleep onset. The importance of sleep consistency, going to bed and waking at the same time every day, including weekends, for maintaining deep sleep architecture is increasingly emphasized by sleep researchers. The body's circadian clock is entrained by consistent timing. And this entrainment is what allows the deep sleep window to be reliably available at the right time each night. Social jet lag, a term coined by chronobiologist Till Roenberg to describe the mismatch between a person's biological clock and their social schedule, is associated with reduced slow-wave sleep quality. Even in people who obtain adequate total sleep duration, the research consistently suggests that regularity of sleep timing is one of the most modifiable factors in improving sleep architecture, and one of the most commonly overlooked. A consistent bedtime does not simply feel more restful, it biologically is more restful. Temperature-assisted sleep enhancement has moved from basic science into research application in recent years. Devices that circulate water at controlled temperatures through a mattress pad have been studied in clinical settings, with research finding that cooling the sleeping surface can increase the time spent in slow-wave sleep. A study published in the Journal of Sleep Research found that participants using a cooled sleeping surface showed electroencephalographic evidence of deeper sleep, with increased slow-wave activity compared to nights without temperature assistance. The approach is consistent with what basic sleep science predicts, and while it is not a solution for underlying sleep disorders, it represents a low-risk method of supporting the body's own temperature regulation mechanisms during sleep. The principle is simple. When the environment helps the body do what it is already trying to do, deep sleep comes more easily. The concept of sleep banking, attempting to accumulate extra sleep before anticipated sleep deprivation, has been explored in research, with mixed conclusions regarding deep sleep specifically. Some studies have found that extended sleep in the nights before acute sleep deprivation can partially buffer cognitive impairment during the deprivation period. But slow-wave sleep, being largely governed by the homeostatic pressure that builds during waking, cannot simply be pre-accumulated by sleeping extra when the pressure is not there. The body generates deep sleep in response to need. It does not store it in advance in any meaningful sense. The relevance of this for daily life is that catching up on sleep after a period of deprivation is possible to a degree. But the idea of banking deep sleep for future use does not have a strong biological basis. The body lives in the present. so does deep sleep. The total light environment of the evening, not just screens, but ambient room lighting, influences melatonin onset timing and therefore the availability of deep sleep at the biologically appropriate time. Dimming room lights to 10 lux or less in the 2 hours before desired sleep onset has been shown to accelerate melatonin onset compared to standard indoor lighting levels of 100 to 500 lux. This seemingly small behavioral adjustment, choosing dimmer lighting in the evening, has measurable effects on sleep architecture. The body's light-sensing system evolved in a world where the only evening light came from fire. Warm, dim, and flickering, modern indoor lighting, by contrast, signals midday to the circadian clock. Every hour spent under bright artificial light in the evening is an hour spent gently telling the body it is not yet time for deep sleep. Wearable sleep technology has made it possible for millions of people to monitor their sleep outside of a laboratory. Devices that track movement, heart rate variability, and skin temperature can provide estimates of sleep stages, including estimates of deep sleep duration. The accuracy of consumer devices in distinguishing slow-wave sleep from other sleep stages has improved substantially over the past decade. Though clinical polysomnography, the gold standard involving brainwave measurement, remains significantly more precise. Research evaluating consumer devices has found that they tend to underestimate the time spent in lighter, non-REM sleep and may overestimate time in deep sleep in some conditions. Nevertheless, the trend data they provide, showing how sleep architecture changes in response to lifestyle factors over time, has value for people seeking to understand their own sleep patterns, provided the limitations of the technology are understood. The concept of slow-wave sleep deprivation, as distinct from total sleep deprivation, has been studied through selective suppression experiments, where participants sleep normal hours but are awakened each time slow-wave activity begins, without reducing their total sleep time. These studies consistently find that selective slow-wave deprivation produces metabolic impairment, reduced immune function, increased pain sensitivity, and next-day fatigue, even when total sleep hours are preserved. This finding has important implications for understanding the sleep complaints of people who sleep long hours but feel unrefreshed. The issue may not be quantity, but architecture. A night of 8 hours in which slow-wave sleep is repeatedly fragmented may produce the same functional deficits as a night of considerably shorter but architecturally intact sleep. Hours in bed and hours of genuine deep sleep are not the same thing, and only one of them restores the body. The neurobiological mechanisms of deep sleep continue to be revised as research methods improve. A significant recent development has been the use of optogenetics, a technique allowing researchers to switch specific neurons on or off using light to identify the precise cellular populations responsible for slow-wave sleep generation. Studies using optogenetic tools have identified specific interneuron populations in the cortex that appear critical for initiating and sustaining slow oscillations. These same populations show age-related changes consistent with the reduction in slow-wave sleep seen in older animals and humans. The identification of precise cellular targets opens the possibility of pharmacological or neurostimulation interventions that could selectively enhance slow-wave sleep, a possibility that represents one of the most actively pursued goals in current sleep medicine research. Science is learning not just why deep sleep is disrupted, but how to help the brain find its way back to it. We have arrived at the answer the science has been building toward. The question, why can't you stay in deep sleep long enough? turns out not to have a single answer. It has many answers, layered on top of each other. Deep sleep is the most physiologically demanding state your body attempts each night. It requires precise temperature conditions, the right hormonal balance, a quiet arousal system, a well-timed circadian clock, an open airway, a brain that has been allowed to wind down, and a body whose stress response is not still firing from the day before. Most people are not dealing with one disruption. They are navigating several at once, layered, interacting, and often invisible. The good news, embedded in this same science, is that most of these factors are not fixed. They can be changed, and when even a few of them change, the body moves, sometimes quickly, sometimes gradually, back toward the depth of sleep it has always been trying to reach. You might be feeling tired now. That's perfectly fine. Breathe slowly. Feel the cycles. Your heart has beaten more than 2 billion times without a single reminder from you. Your lungs have breathed without instruction. Your cells have divided, repaired, renewed, all without your help. Your body knows how to take care of itself. Tonight is one more instance of that ancient, patient knowledge. You don't need to hold on to anything. Simply let the cycle turn. We're almost there now. Deep sleep does not require effort. The harder you try to sleep, the more the arousal systems of the brain activate, making deep sleep less likely, not more. This is one of the central paradoxes of sleep, and one of the most practically important things sleep science has established. The state most hostile to deep sleep is anxious effort. The state most compatible with it is quiet allowing. What the body needs is not motivation. It needs permission. The brain during deep sleep is more disconnected from the outside world than during any other state of natural consciousness. Sensory signals are still received and evaluated. The thalamic gating mechanisms remain active, but the threshold for what reaches conscious processing rises dramatically. The world continues. Sounds continue. The night continues. The sleeping brain simply chooses, with extraordinary selectivity, what deserves its attention. Most things, it decides, can wait until morning. Slow-wave sleep is associated with a reduction in activity across the brain's default mode network, the system involved in self-referential thinking, worry, and rumination. The quieter this network becomes, the deeper the sleep. This is perhaps why the most reliably sleep-inducing experience is one of gentle, effortless engagement with something outside the self. A story, a voice, a series of facts that drift through without demanding judgment or decision. The self, briefly, becomes less important than what is simply present. The body heals its tissues most effectively during deep sleep. Microtears in muscle fibers, caused by exercise or daily activity, are repaired. Bone mineral density is maintained through overnight hormonal processes. The skin's barrier function is strengthened. The lining of the gut renews itself. None of this happens all at once, and none of it requires awareness. It is the body's long, patient work, done in the quietest hours, completed before the first light. Children who obtain adequate deep sleep show measurably better emotional regulation during the following day. Research with school-age children has found that nights with more consolidated slow-wave sleep predict lower rates of emotional reactivity, better frustration tolerance, and fewer behavioral difficulties across the subsequent day. The relationship holds even after controlling for total sleep time. it is the architecture that matters. Not only the duration, the same pattern is present in adults. Deep sleep is, in one of its many roles, a nightly recalibration of the emotional thermostat, the sound of slow, steady breathing, whether your own or someone nearby, carries a biological signal associated with safety. Research on the acoustic environment of sleep has noted that certain sound patterns, including rhythmic breathing sounds and low-frequency noise consistent with natural environments, tend to be associated with more stable sleep architecture. The brain appears to have a preferential response to patterns that signal the presence of safe, sleeping companions, an evolutionary inheritance from the millions of years during which humans slept in groups. The sound of this voice, perhaps, carries something of that quality in its rhythm. Magnesium plays a quiet but meaningful role in supporting slow-wave sleep. It acts as a natural regulator of the NMDA receptor, a glutamate receptor involved in neural excitability, blocking it at resting membrane potential and reducing the likelihood of spontaneous neural activity. By moderating cortical excitability, adequate magnesium levels may support the conditions necessary for slow-wave oscillations to form and be sustained. Dietary surveys in the United States consistently find that a substantial proportion of adults consume less magnesium than recommended. Foods rich in magnesium, leafy greens, pumpkin seeds, almonds, black beans, are among the most quietly supportive additions to a diet that is trying to sleep more deeply. The concept of sleep as medicine is gaining traction in clinical settings. Increasingly, sleep assessment and sleep quality optimization are being incorporated into the management of conditions as diverse as diabetes, depression, chronic pain, cardiovascular disease, and immune disorders. The recognition that improving sleep architecture, not just extending sleep duration, but improving the depth and quality of the sleep obtained, can produce measurable improvements in disease management, represents a meaningful shift in how medicine views this most fundamental of biological needs. Sleep is no longer a side note in clinical care. It is becoming a treatment. Even a single night of improved deep sleep can produce measurable changes the following day. Studies tracking biomarkers before and after nights of high versus low slow-wave sleep find differences in inflammatory markers, in glucose metabolism, in cognitive performance, and in emotional reactivity. All within 24 hours. The body responds quickly to the opportunity to restore itself. It does not need weeks of perfect sleep to begin feeling the difference. It needs one night that is a little quieter, a little cooler, a little less interrupted, and then another. The architecture of deep sleep reflects the architecture of the life lived before it. A day spent in physical activity, in social connection, in purposeful engagement with the world, with adequate light in the morning and dimming light in the evening, with meals at consistent times and a body that has been respected. Such a day creates the biological conditions from which deep sleep most naturally grows. Sleep is not separate from the day. It is its completion. The thalamus, described earlier as a sensory gatekeeper, is also something like a quiet companion during deep sleep. While the cortex sweeps through its slow oscillations, the thalamus maintains its own rhythms. The sleep spindles and K-complexes that punctuate the deeper stages of non-REM sleep. These thalamic signals may serve to both protect the cortex's slow waves from disruption and to facilitate the memory transfers that make deep sleep so essential for learning. the thalamus and cortex, in their overnight dialogue of waves and rhythms, are doing something together that neither could do alone. The experience of waking from deep sleep, that heavy, reluctant return from somewhere very far away, is one of the more distinctly physical sensations the body produces. The weight in the limbs, the slight confusion about time and place, the way the room seems to need a moment to reassemble itself. This is what genuine slow-wave sleep feels like from the inside of the return journey. It is, in its own way, evidence, felt evidence, that the body had gone somewhere real during the night, somewhere it needed to go. Your brain's electrical activity during the deepest moments of slow-wave sleep resembles, in some studies, the activity recorded in states of deep meditation, particularly the increased delta wave power associated with certain forms of contemplative practice. Some researchers have explored whether meditation training can directly enhance slow-wave sleep quality, with early findings suggesting that long-term meditators show more consolidated deep sleep and higher slow-wave amplitude compared to non-meditating control subjects. The connection between the interior practice of quieting the mind and the biological practice of deep sleep is closer than it might first appear. Both are states of profound settling. Both require, in the end, the same thing: the willingness to let the activity stop. The hours between 10 in the evening and 2 in the morning are, for most people following a natural sleep schedule, the window during which slow-wave sleep is most available and most abundant. This is when the circadian clock and homeostatic sleep pressure most strongly converge, creating the optimal conditions for deep sleep. People who are awake during these hours, by choice or by circumstance, are typically foregoing the majority of their slow-wave sleep for that night. Regardless of how long they sleep afterward, the body will shift toward REM-dominant sleep in the later morning hours. But the deep sleep window, for most people, is found in the earlier part of the night. Staying in deep sleep long enough begins, most often, with being asleep at the right time. We have now traveled through nearly 100 facts about why deep sleep is interrupted. And the answer, looked at from this far along, is simpler than the science might suggest. The body knows how to sleep deeply. It has always known. What it struggles against is the accumulation of small conditions that were never designed to coexist with it. The light that is too bright, the schedule that is too irregular, the substance that blocks the signal, the thought that will not release. None of these are permanent. Each one is something that, understood, can be gently addressed. And when it is, the deep sleep that was always waiting finds its way through. Children awakened from deep sleep are often so deeply unconscious that they can be moved, spoken to, or even walked around without waking. A phenomenon parents of young children know well. This deep unconsciousness reflects the intensity of slow-wave activity in young brains. It is also one of the mechanisms behind sleepwalking and sleep terrors, which occur in the first third of the night during slow-wave sleep episodes, and involve partial arousal states in which motor circuits become active while consciousness remains in the deep sleep state. These parasomnias are generally harmless in children and most commonly outgrown. Evidence of a developing system still calibrating the boundary between deep sleep and wakefulness. Slow-wave sleep appears to be particularly important for consolidating procedural memories, the kind involved in physical skills, studies examining the learning of motor sequences, finger-tapping patterns, musical scales, surgical techniques, have consistently found that slow-wave sleep in the hours after training is associated with better performance at the next test session. Every physical skill, every movement learned and practiced, is being refined in the quiet dark by the slow waves that pass across a sleeping brain. When deep sleep is cut short, this refinement is interrupted. The skill is not lost, but it is not as fully made. The role of the astrocyte, the star-shaped support cell of the brain, in sleep regulation has grown considerably in scientific understanding over the past decade. Astrocytes are now known to be active participants in the regulation of sleep need. They release adenosine over the course of wakefulness, contributing to the accumulation of sleep pressure. They also play a central role in glymphatic fluid dynamics, forming part of the channel network through which cerebrospinal fluid moves during deep sleep. And recent research has found that they participate in the synchronization of slow cortical oscillations, not merely passively supporting neurons, but actively contributing to the rhythms of deep sleep. The sleeping brain is not simply neurons, it is an ecosystem, and the support cells are as essential as the stars they are named for. The experience of deep sleep. From the outside, watching another person sleep deeply is one of the most visibly peaceful states in human experience. The face relaxes in ways it rarely does when awake. The forehead smooths. The jaw settles. The small muscles around the eyes go quiet. The breath becomes slow and regular. The person seems not merely to be resting, but to be somewhere else entirely, held in a state so complete that the ordinary concerns of the waking world have, for this time, genuinely ceased to exist. This is not an appearance. It is what is actually happening. The concerns have not paused. They have been set aside, not by will, but by biology, by the deep and ancient mechanism of sleep. The science of deep sleep continues to unfold. Each year, new tools, more sensitive neuroimaging, more precise optogenetics, more comprehensive genomic analysis, larger population studies reveal new layers of what happens in this most fundamental of states. What remains constant across every finding is the same conclusion. The body wants to sleep deeply. It is designed for it, shaped for it by millions of years of evolution. The obstacles to deep sleep are real and many, but they are, in the largest sense, obstacles placed in front of a process that has been running successfully since long before there were names for what it is or why it matters. The body remembers, even when we forget, and tonight, gently, it is trying again. And so, our quiet journey through the science of deep sleep comes to a gentle close. We began with something small and specific, the slow delta wave, sweeping across the cortex at half a cycle per second, and discovered that in that unhurried rhythm, the body performs some of its most essential work, the glymphatic clearance that removes what accumulated during the day, the growth hormone pulse that arrives in the first hour of deep sleep and will not come again until the following night. We learned that the question, "Why can't you stay in deep sleep long enough?" does not have one answer. It has many, layered quietly on top of each other. The light left on too long, the caffeine still circulating at midnight, the stress response still firing from a conversation that ended hours ago. From the first delta wave to the last, we glimpsed how many separate forces are working, each night, toward the same simple goal. That goal is not mystery. It is rest. True, cellular, restorative rest. and it is available to you. Deep sleep reminds us that the most essential things happen in silence. The glymphatic fluid moves without sound. The growth hormone pulse releases without announcement. The slow waves pass across the cortex without a single thought to guide them. The body does its deepest work in the dark, in the quiet, in the state of least interference, by simply allowing those conditions to exist, by dimming the light, lowering the noise, releasing the day's remaining tensions. We offer the body what it has always been trying to reach. Without that offering, the night passes lightly. With it, the body remembers what it has always known how to do. In these final minutes, take the deepest breath you can, as deep as possible. Think about unity of every sleeping mind, in every time zone, in every quiet bedroom across the world. Breathing slowly right now. Think about the slow waves moving across their cortexes and yours. The same rhythm, the same ancient, patient knowledge. You are not separate from that. You are part of it. Let your entire being dissolve into the darkness behind your eyes, into the quiet before the first thought forms, into the space where the body knows, without being told, exactly what to do. Prepare for deep sleep. No boundaries left, only peace. This gentle journey through the science of deep sleep, and every night you return to it, helps other sleepless minds find their way here too. A soft like or subscribe passes it forward quietly. It only takes a moment, one quiet tap, one sleepy soul at a time. But there's no need to do anything now. Nothing at all. You've traveled far tonight, through the quiet science of what happens when the body finally lets go. You can allow these thoughts of delta waves and slow oscillations to fade now, like the last edge of light before a room goes completely dark, leaving only the still, warm calm behind. Let your breathing deepen. Let your body feel heavier, as if something patient and vast is gently holding you, the way the earth holds everything that rests on its surface, without effort, without interruption. If sleep comes, let it carry you the way a slow wave moves across the cortex, not rushing, not falling, simply arriving into a realm where time and weight no longer matter. If your eyes are still open, the next video is waiting for you, ready to continue the journey. But if they're closed, stay there. Stay in the darkness that feels safe and warm. Tomorrow, the science will still be there. The slow waves will still be waiting for you, still moving, still clearing, still doing their patient work. But tonight belongs to stillness. Rest well, sleepy soul, and remember, the deepest sleep begins the moment you stop trying to reach it. Good night.
you